The topic of today's discussion is neuronal transmission, that is the process by which the transmission of information takes place through the nervous system. What we see in this figure is our nervous system, which is constituted of both the central nervous system, that is the brain and the spinal cord, and the peripheral nervous system. And this is actually a network of specialized cells called neurons that transmit information from one part of the brain or body to another. Now, this information can be sensory in nature, that is received from the outside world through sight or hearing, and then it is processed by the brain and then transmitted to the muscles via the motor neuron, where it is converted into action. Therefore, this complex network of neurons is responsible for all our thoughts and actions, and the information is transmitted through this network by the process of both chemical and electrical transmission. Now, this figure shows a diagram, a schematic diagram of two neurons which are placed in tandem. The gap between the two neurons is called the synapse. The neuron which lies before the synapse is called we will call the presynaptic neuron, and what is after the synapse is called the postsynaptic neuron. The neuron has, as is seen in this figure, is a polarized cell which has a distinct morphological regions which are responsible for specific functions. The information is transmitted along this neuron by electrical transmission, whereas information is transmitted between the neurons, that is from one neuron to another, by chemical transmission. This figure also indicates the unidirectional nature of this electrical transmission from one part of the neuron to another and from one neuron to the next neuron. One should mention at this point that this is a very simplified diagram in which we see that one neuron is interacting with only one and another neuron, but in reality, one neuron can interact with up to 10,000 other neurons, which therefore shows the complexity of the network which will be created using such a system which is present in our brains. Now, in this figure, we shall see the morphological features of the neurons. First, we have the cell body or soma, which contains the nucleus and all other organelles, which is responsible for cellular function. Projecting out of the cell body are these dendrites, which connect with the previous neurons and receive information from the previous neuron. This information is then transmitted along this elongated region of the neuron, which is called the axon. By this rapid changes in membrane potential, which travels along this axon, this information then reaches the synapse, which is the terminal region of the axon, when it is conveyed to the next postsynaptic neuron. But now, during this communication between the presynaptic to the postsynaptic neuron, this communication takes place via chemical transmission. Now, the neurotransmitter, which is the chemical messenger, is packaged inside the vesicles. When this action potential, which travels along the axon of the neuron, this nerve impulse reaches this terminus of the presynaptic neuron, the vesicles packaged with the neurotransmitters fuse with this membrane, releasing the neurotransmitter to the synaptic cleft. The neurotransmitters then sit on the receptors which are present in the postsynaptic membrane. This leads to certain changes, like changes in membrane permeability of the postsynaptic membrane, which leads to an action potential generated in the postsynaptic cell, and therefore the information is transmitted from one neuron to another. Now, in order to understand how the electrical stimulus is generated and passes from one end of the axon to another, we have to appreciate that there are specialized specific ion channels which are present on the membrane of the neuronal cells. And these channels can be classified as chemical gated or ligand gated ion channels, which open when a specific ligand binds to these channels, and this allows the movement of specific ions across the membrane. Or there might be voltage gated ion channels which open only when there is a change in the membrane potential. This brings us to the concept of resting membrane potential. All the cell membrane maintains a resting membrane potential with the inside more negative than the outside. That is, the cytosolic side is more negative than the extracellular side. And this membrane difference in electric charge across the membrane can also be modeled as a capacitor. How is this resting membrane potential generated and how is it maintained? The resting membrane potential is generated due to the action of these potassium channels and sodium channels and the sodium potassium pump which is present on the membrane. The K plus ion channels leads to the loss of K plus ion through the leak channels from the inside of the cell to the outside. One has to remember that all these ions are present along with their counter ions, for example, Cl minus ion. The movement of K plus ion selectively across the membrane leaves an excess of Cl minus ions in the inside of the cell. Therefore, the interior of the membrane becomes more negative. However, with the with the movement of the Na plus ion channel through the Na plus leakage channels, this reduces the negative membrane potential slightly because the Na plus ion is moving from the outside to the inside. In addition, there is the sodium potassium pump, which pumps for every cycle 3 Na plus ion outside the cell for every 2 K plus ion which is pumped inside the cell. And it is due to this joint action of the K plus leak channel, Na plus leak channel and the sodium potassium ion pump, we have a resting membrane potential of approximately minus 70 millivolt, which is generated across the cell membrane of all cells. The question is, can we measure experimentally resting membrane potential? Yes, in this experiment, we use a microelectrode, which is a small piece of glass capillary tubing with a very fine tip, and this tubing is filled with a conducting solution and attached to a recording device. When this electrode is placed in the extracellular medium, since the extracellular medium is isopotential, therefore no voltage is recorded. However, as soon as we insert this electrode tip into the neuronal cell, a potential of about minus 60 millivolt with the inside negative with respect to the outside is recorded, and this is called the resting potential. The potential is constant for indefinite periods of time. All membranes have this resting membrane potential. What is unique for the nerve cells and other excitable membranes like the muscle cell is the resting membrane potential of these cells are capable of changing. Now, can we detect an action potential in a neuron? Yes, for this we insert a recording electrode and a stimulating electrode. The stimulating microelectrode actually passes a negative or a positive current in order to stimulate the nerve cell. The change in the membrane potential which results from this stimulation is recorded via the recording electrode. Now, if we look at one action potential, that is one such spike, we can divide this spike into 4 distinct stages. That is the resting state, the depolarization state, the repolarization state, and the hyperpolarization state. Now, in this resting state, the membrane potential is maintained at the resting membrane potential. Now, upon stimulation, depolarization is initiated and there is a decrease in membrane potential which changes towards the positive until it reaches approximately plus 30 to plus 40 millivolt. This is the depolarization phase. This is followed by the repolarization phase when the membrane potential again changes towards the negative resting membrane potential, which is then followed by the hyperpolarization phase where the membrane potential becomes more negative than the resting membrane potential. Eventually, an equilibrium is reached when the resting membrane potential of minus 70 millivolt is again restored. Now, what leads to these different phases of action potential? Now, in the initial resting state, all the voltage-gated sodium and potassium ion channels are closed. Now, upon depolarization or stimulation, what happens is the Na plus ion channels open, which allows the entry of Na plus ions from the outside of the cell to the inside, which leads to a change in membrane potential towards the positive direction. Once the plus 30 to plus 40 millivolt is reached, the Na plus channels become inactivated and the K plus ion channels open. This leads to the transport of K plus ions from the inside of the cell to the outside, and this leads to the change in the membrane potential towards the negative. The K plus ion channels, however, remain persistently open for a long period of time, which leads to this phase of hyperpolarization. But eventually, both the Na plus and the K plus voltage gated ion channels close and the membrane potential is restored to its resting value, which is minus 70 millivolt. Now, can we experimentally detect an action potential? In order to do that, we insert a recording electrode and stimulating electrode inside the nerve cell. The recording electrode measures the membrane potential and the stimulating electrode actually stimulates the neuron and is connected to a battery. As is seen in this figure, initially when the stimulus is low, the depolarization is not strong enough to initiate an action potential. But when the stimulus is high, the depolarization leads to an action potential, which is seen in the form of a rapid depolarization and then a subsequent repolarization phase. This action potential achieves a peak of about plus 40 millivolt and it persists for approximately 0.5 milliseconds, each of these action potentials. Now, how do these action potentials move in one direction along the axon? In order to understand that, we look at this figure, which shows that if we stimulate this portion of the axon, the Na plus ion channels, which are present locally, opens in response to the stimulus. Now, therefore, a depolarization is initiated in this point A. This depolarization also spreads to point B, which now leads to the opening of the sodium ion channels in point B. And an action potential is now starting to be generated at point B. By this time, the repolarization phase has started in point A, where there is the inactivation of the sodium ion channel and the opening of the K plus ion channel. The action potential has now moved on to point B. And subsequently, this depolarization in point B leads to the spread of depolarization in point C, and the action potential now moves to point C, and this is how the action potential moves unidirectionally from one point to another along an axon. Now, many of the neurons which are present in our nervous system are myelinated. In these myelinated neurons, the axon of the neuron, which is the elongated portion on which the electrical stimulation passes from the cell body to the synaptic terminus, the axon is actually covered by a membranous sheath which is called the myelin sheath. This myelin sheath actually provides an insulation across on the neuron. Now what happens is all the portions of the axon is not myelinated. There are gaps in present in myelination, and these are called the nodes of Ranvier's. And interestingly, all the voltage gated sodium and ion channels are located at these nodes, which is the gap between the myelination present along the axon. Now, what happens when the action potential travels along a myelinated axon? Now, suppose the action potential is generated at point A, and because the ion channels are voltage gated ion channels are present only in this unmyelinated regions, the action potential is actually jumps from point A to point B to point C. Therefore, the transmission of electrical stimulus along a myelinated axon is much faster than for a non-myelinated neuron. We therefore now summarize what we have discussed till now, that the neurons or the nerve cells are the fundamental units of the brain and the nervous system. The neurons communicate through both electrical and chemical signals. The electrical signals carry information along the axon of a neuronal cell in the form of action potential. The action potential or nerve impulses are generated by the rapid and coordinated opening and closing of the voltage-gated sodium and potassium ion channels which are present on the neuronal membrane. And the action potential travels from the cell body to the synapse, and myelination of the neuron increases the speed of this neuronal transmission.